I've covered chemistry, I've covered physics, and now let's take it one step further and do biology. Starting off with the basics, we have cells, right? Now, you have two types of cells, prokaryotic cells and eukaryotic cells. Now, eukaryotic cells are the more complex cells, like animal cells and plant cells, for example. I will put a diagram of a eukaryotic cell up right now so you can see what it looks like. Moving on to prokaryotic cells, these are simpler cells, and more often than not they can be bacteria and stuff like that. You know, as I said, simple cells. They don't have a nucleus, so DNA is stored in a completely different system. I'll put up a graph so you can... A graph? I didn't mean graph, I meant image. I'll put up a diagram. That's what I meant, not graph. Speaking of cells, you need to look at them in biology, and that's where microscopes come in. There are things like light microscopes, which literally you put a slide in, the light shines through it and you look through it like that. Those are probably the ones you will use in school. However, you can get things like electron microscopes, which are like really powerful microscopes that you can get massive detail in the objects that they're looking at. It's actually quite impressive. In fact, even images like sugar under an electron microscope is really interesting. You can even view like loads of different bacteria under an electron microscope. It does get very fascinating. With microscopes, as I said, light microscopes, you have a slide, and you also have to prepare these slides. So, with the AQA course, you have to know how to prepare a slide. Now, the steps goes as follows. 1, you add a drop of water onto the slide. Then, you grab your specimen, whether it's a bit of onion or a leaf or whatever, you cut that up into the appropriate size, and make sure you can actually fit it onto the slide. Next, you need to add a drop of iodine solution. Now, what this does is it dyes your specimen, and it kind of highlights everything in the specimen, as it were, so you can see it through the microscope a lot better. It's kind of a way of, as I said, highlighting the things that you're looking at. Now, once you have that dyed specimen on your slide and it's all prepared to go, you then put that into the microscope and then adjust this and that, and then look through it, and boom, you have your stuff. Now, sticking with cells, you have specialized cells. Now, specialized cells are cells that are specialized. It's there in the name. Now, these cells, as I said, they're specialized to do certain tasks, right? So they will have these different adaptations to them to make them better at their job. Some of the examples I have are things like the root hair cell, or the muscle cells, or the nerve cell. And some of the adaptations, as I said, like for the nerve cell, you have, it's kind of spread out, it's big. Put up a picture of a nerve cell so you can see what it looks like. And actually, the other cells you need to know for the AQA course, because you need to know these specialised cells. That's nerve cell, muscle cell, phloem and xylem cell, sperm cell, and muscle cell. Wait, did I say muscle cell? Root hair cell, that's what I meant. But those are the ones that you need to know, okay? And they will have these different adaptations to make them better at their job. Now, in cells, you have a nucleus, right? And in that is DNA. And DNA is stored by chromosomes, or something along those lines. Now, chromosomes, not chromosome. I'm tired. Cut me some slack, okay? Chromosomes. Now, you've probably heard of these before, okay? Chromosomes contain genetic information. You know, coiled up lengths of DNA. And each carry a large number of genes. Now, genes basically tell you what colour your hair are, what colour your eyes are, how tall you'll grow, etc, etc, etc. Body cells usually have 2 sets of chromosomes, which is one from the mother and one from the father. Now, moving on to mitosis, right? Mitosis is basically cell division. You know, cells multiplied by dividing. Yeah. But yeah, mitosis is basically the way cells go from this to this to this. It is important to know that prokaryotic cells, however, use binary fission, not mitosis. Now, you can actually grow your own bacteria and your own microorganisms and all that stuff using a culture medium. Now, what a culture medium is basically something you can grow the bacteria on. It contains basic carbohydrates and nutrients and all that stuff. Now, the most commonly used one is agar jelly. You know, it's a weird, it's kind of like a weird greyish-brown colour, and it's partly see-through. You've probably seen it before, or heard of it at least. That's what you use to grow bacteria on. Now, it's important to note that when you're growing bacteria, you should always store it below 25 degrees Celsius, otherwise harmful bacteria could grow instead of normal bacteria. And you don't want that to happen, otherwise, worst case scenario, you die. Now, you can grow bacteria and you can use antibiotics to see the effect on growing bacteria. So when you're growing the bacteria, you put the antibiotics onto whether it's a piece of paper or actually just in the jelly itself, depending on what you're doing and what the antibiotic is. When the bacteria grows, you can then see how it is affected, and you can see how effective the antibiotic is by how big the radius is around the antibiotic and like where the bacteria is and stuff like that. Now, when it comes to growing bacteria, it's actually important to note that you should always keep the Petri dishes, that's what you grow the bacteria in if you didn't know, stored in a specific way. They need to be sterile when you're doing the test, so you don't get any alien bacteria or any, well, I say alien, I don't mean alien from another world, I mean like bacteria that you don't want in the Petri dish, okay? So probably foreign bacteria would probably be better terminology, but you don't want any foreign bacteria getting into the Petri dish, so you need a sterile area. Often flames can help sterilize equipment because you just heat it up and it kills the bacteria on it. And actually, fun fact, that's why sometimes when you use a needle to pry out splinters, and yes, you can use a needle to pry out splinters, you should always heat it up before you do so. Then let it cool down because you're sterilising the needle, so when you use it you don't infect your hand, for example. Anyway, as I said, when you're growing bacteria in a Petri dish you need certain things. You should always have the lid securely on, as I said, to prevent foreign bacteria from entering your experiment. And this, okay, when it comes to the lid, right, you don't want to just put the lid on and wrap a bunch of duct tape around it, because then there's going to be no oxygen going to the Petri dish. And then you'll grow oxygen then you'll grow bacteria in a non-oxygenated environment. It's a bad thing when you grow bacteria in an unoxygenated environment because they can be really deadly sometimes. And when you're storing the Petri dish, it's important that you turn it upside down as well. And you probably think, why the hell would you turn it upside down? It's to prevent condensation from dripping onto the actual agar jelly. Because if you have it that way, if condensation forms on the lid, it'll drip down on the jelly and it could alter your experiment. Or if you turn it upside down, the condensation will just stay on the lid and not drip upwards, because that's not how gravity works. This is just starting the biology B1 topic. There's some more stuff after this as well I'll go through. I hope you guys have enjoyed this video. I hope you've learned something. And actually, biology is one of my weakest subjects, I'd say. Or, well, I don't mean to offend any biologists out there, but I do prefer physics and chemistry over biology. It's just a bit more interesting. Sorry. No, but on a serious note, okay, as I said, biology, I don't enjoy it as much. But I'm still going to be pumping out videos for you, one a day. That's what I'm going to try and do. That's what I've been doing for the past couple of days, more than my actual channel. But anyway, I hope you guys have enjoyed this. As I said, I hope you learned something. Remember to leave a like and, help support this video so other people can learn what you learn. And subscribe if you want to watch future videos and learn from those future videos. Hopefully. Bye!